Hello everyone, I am Megha Kulshrestha. Welcome you all on YouTube, and this is my link. I am working as an educator on an academy, so this is the link by which you can follow me on that platform. Okay, so now we will talk about the next class, which is the ascomycetes. In the previous lesson, I discussed with you about the zygomycetes and phycomycetes. Okay guys, so now let's focus on. So this is the classification of fungi, and we had already talked about zygo and phycomycetes with you. Here we will focus on ascomycetes, also known as sect fungi. Phycomycetes are known as algal fungi, zygomycetes conjugation, deuteromycetes fungi imperfecti, basidiomycetes all known as club fungi. Okay, in the phycomycetes and zygomycetes, the mycelium is cenocetic, whereas in ascomycetes and basidiomycetes it is septate. Okay, so let's focus here. So these all are the characteristic features of the class ascomycetes, these sect fungi. Mycelium is septate. Okay, now yeast are an exception in that they are basically unicellular. Okay, yeast are unicellular fungus, whereas the rest other members of the ascomycetes have mycelium structure. Okay, only the yeast is an exception in this. Now the septa in ascomycetes, if you observe the septa contains central pores called the septal pores, also known as dolipore. Okay, so such dolipore like structure you will observe in the ascomycetes. So these are the pores, and sometimes they may get plugged by a body known as Voronin body. Okay, they are plugged by Voronin bodies. Now what is the function of these septa? These septa allow communication between the adjacent cells. Okay, so this is their main function, to allow the communication between the adjacent cells. Now in this class, ascomycetes, yeast reproduce asexually by fission and budding, whereas the majority of the ascomycetes members reproduce through the conidiophores. Okay, their asexual reproduction is through the conidia, which are formed on the conidiophores. Right, conidiophore is a branch-like structure in which many oval shaped conidia are formed, so they reproduce by the conidiophores. Right, now the conidiophores may be branched or unbranched, scattered or aggregated. If they are aggregated, they form some structures. These name of the structures are acervulus, cinema, sporodochium and pycnidium. Learn these names. They form these structures like acervulus, cinema, sporodochium and pycnidium. Okay, so in some of the members of ascomycetes, they form such structures. Now if you talk about the sexual reproduction in ascomycetes, they reproduce through gametic copulation. Example, yeast, it reproduces by gametic copulation, and gametangial contact you will observe in Pyronema, in which anthridium, male anthridium is there and female ascogonium is there, and spermatization in Ascobolus and spermatogamy in Pizziza. So okay, so ascomycetes member reproduce by these methods: gametic copulation, gametangial contact, anthridium by in the gametangial contact anthridium and ascogonium formation occurs, spermatization, and by somatogamy. I hope these things you are aware of. If not, then I'll show you one image. So here you will see this is the gametangial contact. In this way they reproduce. The male, this is the male anthridium and this is the female oogonium. The male forms a fertilization tube. Through this tube, the nucleus of the male enters into the female oogonium. Okay, in the gametangial copulation, these are the strains of different strains of mycelium, plus and minus. They come closer to each other. Their content get fused and they form a structure which is known as zygospore. Okay, now in this case, spermatia formed on the male branch, and this is the trichogyne, this is the female part. Okay, ascogonium, it is known as ascogonium. Spermatia are dispersed and reaches to the trichogyne. This is the trichogyne like structure, and here the fusion occurs and the content gets shared, means nucleus of the male enters into the female part. Now in the somatogamy, there is no formation of such structures. So how the fusion occurs in this? Both the hyphae come closer to each other, and these hyphae are of opposite mating type. One is of plus and one is of minus strain. They come closer to each other, and in between these somatic cells the fusion occurs. Okay, now this cell becomes dikaryon. Two nucleus come closer to each other. So these all are the different way of sexual reproduction in the class Ascomycetes. Now fertilization occurs in two steps in this: plasmogamy and then karyogamy. Karyogamy is delayed after the plasmogamy. Okay, there is a sequence in the fertilization: plasmogamy, then karyogamy, then meiosis. But in the Ascomycetes, plasmogamy occurs, means cytoplasm, fusion of cytoplasm occurs, but karyogamy is delayed. Now there is an intermediate between plasmogamy and karyogamy, that is known as dikaryophase. Okay, that is known as dikaryophase. Now what happens in this phase? After the plasmogamy, what it result in bringing the two compatible nuclei closer. Okay, two nuclei come closer means N plus N. A cell is formed in which two nuclei are there and they are not fused right now, means two nuclei are there but they are not fused. So this is condition in which nuclei are not fused but the cell contains two nucleus is known as dikaryophase. Right guys? Now some dikaryotic cells functions as Eschers mother cell. Some of the dikaryotic cell which is having N plus N, they function as Eschers mother cell. Now in this cells karyogamy occurs, means fusion of these nuclei occurs. Now after the fusion meiosis takes place. Result means reductional division. Meiosis is the reductional division. Then mitosis takes place. Now after meiosis and mitosis, 4 to 8 haploid meiospores are formed in a structure which is known as now Eschers. Okay, Eschers like structure, 4 to 8 cells are formed and these cells are known as ascospores. They are known as ascospores. So ascospores are formed inside the Eschers. Okay, singular is Eschers and plural is Eschai. Now Eschai may occur freely or get aggregated with dikaryotic mycelium. Okay, if they get aggregated with the dikaryotic mycelium, they form fruity fractions, means fruiting body are formed. Okay, and these fruiting body are known as ascocarps. What they are known as? They are known as ascocarps. And the cup-like fruiting body is known as apothecium. Example is Peziza. Flask-shaped perithecium. Example Neurospora. And closed is Cleistothecium. Example is Penicillium. Right guys? So what are the names? Apothecium, cup-like. Perithecium, flask-shaped. And Cleistothecium is closed. So one question can come from this. Okay, so learn these fruiting bodies, names of these fruiting bodies. Let's see the image. Here you will observe Cleistothecium is a closed, perithecium is a flask shape, small opening is there, that is known as ostia, and this is a cup shaped fruiting body that is known as apothecium. So these are the Eschai. Eschai are intermingled with sterile hyphae, they are known as paraphyses. Okay, so they are intermingled with paraphyses like structure. Hope it is clear. Let move ahead now. Now see in this picture, here you will see sexual reproduction. Now what you are able to observe here, so these are the different mating types of hyphae which come closer to each other. Okay, and they come closer to either plasmogamy occurs. Now after plasmogamy, karyogamy is delayed, already told you. So both the nucleus are there in the cell. Now one of the cell behaves as the Eschers mother cell, inside which two nuclei are there, they get fused. Now karyogamy occurs, then meiosis occurs. Karyogamy, after karyogamy, zygote is formed. Zygote undergo meiosis division, 4 nuclei are formed, then they undergo mitosis, they form 8 nuclei which give rise to ascospore like structures. Okay, 8 nuclei will form ascospores. Now these ascospores are now released in the air and they form a new mycelium when they get the suitable environment. Right guys, now move ahead. Now here are the examples of Ascomycetes. Yeast. Yeast is a member of Ascomycetes and it is having a pseudocoelom. It multiply by budding or fission. Okay, no fruiting bodies are there in yeast. Now there are three types of yeast: budding yeast known as Saccharomyces, fission yeast in which fission occurs, Schizosaccharomyces, and both budding and fission occurs in Saccharomycoides. Okay, so these all are the different types of fungi: Saccharomyces, budding yeast, Schizosaccharomyces, fission yeast, and Saccharomycoides, both in which both budding and fission occurs. Right. Now about the Aspergillus. Aspergillus is commonly called green smoky mold. This Aspergillus contaminates laboratory culture and hence it is also known as weed of the laboratory, and it also contaminates various foodstuffs like bread, butter, cheese, syrups, jams, jellies, textile and leather goods. So it contaminates so many things. Aspergillus fumigatus is there, which causes a disease aspergillosis. Okay, Aspergillus fumigatus causes aspergillosis. Aspergillus oryza is used in the preparation of alcoholic beverages. Aspergillus niger is used in the preparation of citric acid. Okay, Aspergillus oryzae is used in the preparation of alcoholic beverages. Aspergillus niger used in citric acid preparation. Aspergillus fumigatus causes disease aspergillosis. Now next is the penicillium. Penicillium chrysogenum is used to obtain an antibiotic that is known as penicillin. You know that Alexander Fleming was the first scientist who has discovered this antibiotic penicillin from the fungus Penicillium notatum. Okay, earlier it was derived from the Penicillium notatum, but nowadays penicillin antibiotic is derived from Penicillium chrysogenum. Remember this, just learn this name, Penicillium chrysogenum. Next member is Neurospora. Neurospora is also known as pink bread mold because of its colors. Okay, now Neurospora crassa is often employed in the studies concluded in experimental genetics. So it is used in laboratories. That's why it is also known as Drosophila of the plant kingdom. Okay, that's why it is known as Drosophila of the plant kingdom. You have heard the scientist named Beadle and Tartan. He has done the experiment on Neurospora and given the conclusion about the one gene one enzyme theory. Okay, one gene enzyme theory concept is given by Beadle and Tartan by working on this fungus Neurospora crassa. Next is the Erysiphe. This fungus produces powdery mildew. Erysiphe graminis causes powdery mildew on wheat and barley, means it causes a disease on cereals, whereas Erysiphe polygoni, it causes a disease on legumes like pea. Okay, next is about the Claviceps. Claviceps purpurea is the common genus of this member, and it produces a disease called ergot of eye. And if such types of cereals are eaten, then it causes a disease called ergotism. Okay, it causes a disease called ergotism, which results in paralysis or in hallucinations or simply the death of that person. Okay, ergotism is caused by which fungi? Claviceps purpurea. Let's move on. Pizziza. It is known as cup fungi because it produces cup-like fruiting bodies. Because of this shape, it is known as cup fungi. Pizziza is known as cup fungi. Now from this group Ascomycetes, there are two members, morels and truffles. They are eaten as foods. That's why they are considered as the edible fungus. Okay, morels and the truffles are known as the edible fungi or edible fungus. So this is all about Ascomycetes. Hope you like this lesson. Please share as much as you can if you found it helpful. Thank you.